Okay. Okay, yeah, that's good. I'm just waiting for a few more people. Hopefully they'll show up. I sent out an email. I don't know. I'm doing all right. I know we saw each other Saturday, but I haven't changed much since then. But I'm doing okay. So let's see here. There's Richard.
Hello.
Hi, Dick. How are you?
Pretty good. Waking up.
Yeah. Oh, Jesse. Yeah, I said I saw you Saturday, but I haven't changed much since then. I see Jesse on Saturday mornings too in a different meeting. So.
Oh, okay.
Yeah. So we're just waiting for a few more people, maybe.
Okay. No problem.
All right. Hello, Yash.
Hi,
How are you? Do you know if Devansh is going to be here?
Yes, he's going to be here.
Okay, just wondering.
Asking.
Hello, Ujjwal. How are you?
Hello, hi, Studio. I'm fine.
Hi.
How are you?
All right. Well, I guess we can get started. And more people come. I'll just let them in. Welcome to this new meeting of the new year. This first meeting. I just wanted to go over actually some stuff from just a recap of DevoWorm for the last year and then also for the last 5 years, because I noticed that it's coming up on sort of I don't know if it's a 6 year, 5 year, or 6 year anniversary of when the group started. So I'm going to go back and look at some of that and then look at what happened in the last year and then maybe some things maybe that will be happening this year. So we'll go through that. I also think I mentioned I have a bunch of people who want to do things in meeting, so I'm going to go over like a weekly agenda that we might fill in, scheduling people for different weeks. So if people want to present something, they can pick a date and we can have it, coordinated, and we can share it so that people can, kind of work towards a goal. So I already have Devansh and Yash in scheduled on this. We'll go through it. So let me start with. Let me share my screen first of all. All right. There we go. First of all, are there any questions before we start? Any new news or anything that anyone wanted to bring up right now? Okay. Well, we can talk about that after. Let me get into the recap. Can everyone see my screen? I have it set up here. So let me do the presentation mode. Okay. So usually by the time I get into this, someone comes in. Someone's knocking to come in. So, all right. If that happens, I'll just exit out of this. And so this is the old idea of Father Time handing off the baton to a baby New Year, and we did this in Worms, of course, for this slide. So we're transitioning from 2019 to 2020, and I wanted to go over the accomplishments during the year 2019 because I think it's important to kind of look back on things. So this is actually a look back over the history. Like I said, this is either coming up on the 5th or 6th anniversary of the initiative. And I wanted to go through like a timeline. So this is the first part of the timeline. So in April 2014, Richard and I met Stephen Larson in a meeting. We met up and we discussed doing this initiative, DevoWorm. And Stephen Larson, of course, is the person, sort of one of the founders of OpenWorm. He was one of the founders. There are a couple people who founded the OpenWorm Foundation, as it's now known, and they started with their PhD work. And so Stephen's work was in neuroinformatics, and their goal, they started OpenWorm like back in 2011, and they were trying to understand the C. elegans, just trying to figure out a way to simulate the entire worm, because it's a fairly simple organism to simulate as opposed to some other organisms that we have available to us. Not only is it a small number of cells and a fairly simple nervous system, although it's pretty complicated, it's also very well characterized, and there's a lot of data out there for people to use. And so this is how DevoWorm started. And OpenWorm at the time did not have anything to do with development. And Stephen thought that this would be a good way to incorporate the developmental aspect. So that's where we started. We didn't really start. We kind of started with a goal of, trying to do what the other projects in OpenWorm were doing, which is creating simulations and that sort of thing. But it quickly turned out that we didn't really go in that direction. So we kind of introduced ourselves to the community with a journal club. And at the time, OpenWorm had journal clubs where you would present to the group and they would get a bunch of people online and you would give these presentations. So this is the first presentation, Raising the OpenWorm, which was a very, a broad level overview of the challenges that this project afforded. And then that was well received. So then we wrote this paper, and this is a paper that took kind of a more theoretical view of the project. So this paper is on bioRxiv, and I didn't put the DOI, but it's DevoWorm, and the subtitle involves differentiation waves in computation. So this was back again in 2014. And so if you want to read about our view of the project back then, you can go find that paper. It's on the website under the publications. You should be able to find it. And it's very different from kind of the stuff we're doing now. Maybe not so much, but it did lay out sort of a vision for what to do, and we accomplished some of those things, and other things we did not. So, I mean, I haven't read it in a long time, even myself. So it's good to look back at things like that and reevaluate it. And so, this early part of the project, we had, a couple people. We had Richard, myself, Steve McGrew, who hasn't been around in a while, but he's an inventor, and a couple other people have come and gone out of the group at that time. And then maybe the next year, I think it was May of 2015, I went to Indiana University to their Biocomplexity Institute, and I gave another talk, which is on the website. If you go to I think it's the media section or something, you can find this talk. And again, this was more about the simulation aspect. So at that time, we didn't really have any papers out, but there was a vision, and it was fairly well received, although it would have been more well received if we had some papers out at the time. But I think, that was a good opportunity to get people interested in the project. Then coming up into 2016, we had a number of things going on at once. So we had a couple of papers. This one I worked on with Richard, quantifying mosaic development. This is a nice paper. It's a theoretical paper, but it's also a lot of data science. And we not only looked at C. elegans, but we also looked at C. intestinalis, which is a sea squirt, which is another very common model in developmental biology. We haven't focused on comparative development since then, really, but that's something that we're interested in. So maybe we'll talk about that later. Maybe we'll revisit that in the new year. Also, information isometry. This is a theoretical paper on a technique for looking at trees in development. And so it's on the website. We had another journal club. And so this journal club was sort of foreshadowing some of these papers and their content. So now we have a little bit more flesh on the bones of this initiative by now. And then, of course, we have another person who's collaborated with us, Tom Portages, who came up with this simulation platform called Morphozoic. And we put out a book chapter on that about the same time. And this paper goes through this platform, Morphozoic, which is an open source cellular automata platform that you can use to classify images, classify shape, and things like that. We haven't really done a lot with it since the book chapter. So again, if people are interested in like a challenge, we have Morphozoic available for people to play with if you're interested in cellular automata, or you're interested in doing something interesting with that. We have the paper and the software for people to play with. So and then we haven't really, I think maybe we should revisit that at some point, because it is, I think there's something there left to explore, I think. So, and then the next month after that was published, we had OpenWorm had a organization-wide open house. And so the reason I bring this up is because we had to create a flash talk for this open house. And this was something that was held online around the world. We would have people go in 5 to 7 minute increments and introduce their projects. So those materials actually are all available from OpenWorm. And if you're interested, I can send you a link. It's really a recap of each project. It's a little bit dated now, but if you really want to get a good overview of OpenWorm, this is a good place to go to get this information. We have, like the 5 to 7 minute long talks, which really distill a lot of the things around the projects. We have like papers, which are a little bit dated now, but they give an overview. So, like, in OpenWorm, you have projects like Cybernetic and C302 and Geppetto, and those are all they're simplified in those presentations, like what they involve, what they do, and I think that's a really nice resource for people who are just kind of casually interested in OpenWorm. So we might do that again because it's been like several years since then, but it was definitely a good opportunity to get our project out there again. Then we move now to the next phase of the timeline. This is from November 2016 to April 2019. So you notice there are fewer arrows, and we stopped doing the journal clubs or the meetings that we did in the first timeline. But we've been actively reaching out during this time as well. So I did a postdoc in a lab where I was doing C. elegans biology. And from that, I ended up writing a paper called Evolution in Eggs and Phases. And so this was a experimental evolution paper. So this is a paper where we took real live worms and evolved those worms for 20 to 25 generations and looked at their reproductive capacity. And so it's a little bit different than like the embryogenesis stuff that we were doing before, but this is something that was also, interesting and might be followed up on. We don't have the wet lab anymore, but, it was just an opportunity that I had. But if we ever got the opportunity to do this either like again with real worms or even simulations of evolution, it could be something to follow up on. So that was published in Royal Society Open Science. Then that was probably the end of the publications for a while. Would be another maybe 1 year and a half before we had any more publications. But in that time, there was a workshop in London at the Royal Society called Connectome to Behavior. And this happened in February of 2018. And I didn't put the link there's a blog post I did on this. I don't have a link to it in here, but I can send it to people. It was basically where the Royal Society hosted a workshop where we brought in a bunch of people who were interested in the connectome. And this is associated with OpenWorm. Notice the coloring of the flyer. And I wasn't at the meeting. I was doing some participation remotely. But it was a very good workshop. They didn't really have a lot of, there isn't a lot of evidence of it now. They didn't record the sessions. But it was a very interesting set of sessions. They documented it on Twitter. So I took a lot of the tweets and put them into this blog post so that we had a more permanent record of the meeting. So, if you're interested, I can send you a link to that maybe after in a post-meeting email. But that workshop led to a special issue of Proceedings of the Royal Society B. Oh, I don't have that right. Okay, so this one, don't disregard this title. This isn't the paper. But there's a OpenWorm paper that's in the Proceeding of the Royal Society that is like a recap of OpenWorm. And so it has 18 authors. And we have all of the different projects featured in it. And in that paper, there's a very succinct statement about what DevoWorm was doing circa 2018 or mid-2018. And so if you compare that with the differentiation waves paper back in 2014, you'll see that it developed quite a bit, and that there were actually, at this time, 3 different aims of the project. And so go over those in the paper. But this Royal Society special issue actually is very good because it has a lot of the stuff from OpenWorm and also some stuff from Connectomic, C. Elegans Connectomics in one place. So we had papers from, people doing connectomics, people doing, movement, computational neuroscience and movement. And then we had papers on things like the C302 platform, the cybernetic platform, a general OpenWorm paper, and a couple other papers in there. Again, that paper is available. It's not open access, but we can get it for you if you need it. And then just before this special issue came out, I actually did a presentation at the Finding Your Inner Modeler workshop at Illinois Chicago, and that was a NSF-funded workshop where they wanted to see examples of biology and computation, or biology and data science, that sort of mix, modeling. And I presented on DevoWorm there. I presented on something called embryo networks. And that presentation is on the website in the media tab on the website. It's interesting because it was this idea that we had been working on for a while, and it would be published a couple months later, but it was pretty well received. So, I thought, I first started giving the talk and people didn't really understand what was going on. And then at about maybe 10 minutes into it, people got into it and figured out what was going on. I found that experience to be quite common when I give talks, because I tend to like to give talks on very complicated subjects. So just for people who, just for people who are interested in that. So there's this embryo networks theme that was emerging about this time. Then Richard and I think Tom was an author on some of these papers as well, actually we edited a special issue of Biosystems. And so this was a special issue on computational, theoretical, and experimental approaches to morphogenesis. And this special issue had a really interesting backstory, because we were originally going to do it for a different journal, and then it didn't really work out so well. We couldn't get a lot of papers for it. So in the meantime, one of Richard's colleagues died, and they wanted to do a special issue dedicated in his honor, and so we took our special issue and the people doing this special issue tribute, and we combined them into this special issue here. And so this is a very good special issue of Biosystems. It has a lot of papers in it on embryogenesis, on morphogenesis, on a bunch of different approaches to it: computational, mathematical, experimental. And so it's a really good, again, there's a link to we published, I don't know how many papers in this, 4 in the special issue. So we had 3 papers on C. elegans, I believe, and then one on some Drosophila data that we had on the imaginal disc in Drosophila, which is a developmental structure. And so we had those publications, and so the Embryo Networks is one of those publications. There was a paper on the developmental connectome. There was a paper on the second cybernetic embryo paper. So I know that Jesse's heard of the cybernetic embryo papers. The second one was published in there, and then a paper on Drosophila development. So that was 2018. Then in 2019, and this is where I met Yash and Devansh in the Mozilla Open Leaders project, where we developed something called the DevoWorm OpenWorm curriculum, which is, if you've not seen it, it's on the DevoWorm GitHub, and it's a bunch of different lessons that involve, development. It involves different topics in development, different ways of viewing development, like dynamical systems, and a tutorial on pattern formation. In this program, I learned a lot about open science, and also developed this platform. And it's just it was just meant to be like sort of a framework or a skeleton for further curriculum building. So that's the way it was meant to be at the beginning, but we haven't really revisited it at all. Nevertheless, there's still a lot of good stuff there. So maybe we'll talk about this in a future meeting and kind of reassess where we are with respect to that curriculum. I'm sure people would have ideas of what we can add to it. I talked a little bit with Richard about it when I was in the program, and we did a little bit of back and forth on some of the items, some of the curriculum programming. So we have things like micro credentials or badges. We have tutorials that are in sort of a walkthrough format where you just go through and you learn different things. If you want to know about pattern formation in embryos, you go to this page and you can read about it. But there's probably more to add, and there may be more interactive ways to present this curriculum. And then finally, this is like the last year, and I'll get into the last year and the vision for the future. But, the last year we've been working on some developmental or some educational initiatives, and we're starting to get another raft of papers out. So this is the digital diatom. This is going to be a book chapter. Raising the connectome, which is something that's being worked on for a special issue of a journal, which is a follow-up to the paper in biosystems on the C. elegans connectome, and then this paper data theoretical synthesis, and we'll talk about that in a minute. And so, now that I've presented all this in a very long format, I wanted to give people, just think about this. We won't necessarily talk about it now, but what are some of the future directions that we might take with some of this history? I mean, there's so many things that, you engage in. You might do a paper, you might do a talk, and then it just sits there and it doesn't go anywhere else. Sometimes it does. With this connectome stuff, there might be some more gas in the tank on that. With the C. elegans data, they've gotten a lot of stuff out of it. But then again, there are other things that didn't really go anywhere. We did like one paper or one talk on it, and that was it. So, future directions, that's what we're thinking of. And this is a C. elegans that's just hatched from its egg. So it's full of possibility. So 2019, in terms of education and research, there are three main educational initiatives in 2019. The first one, of course, is this DevoWorm OpenWorm curriculum, and that happened from January to April. This was sponsored by Mozilla Open Leaders. This is a very good program. If you ever get the chance to be involved in the Open Leaders, Mozilla runs these programs maybe twice or three times a year. And they teach you how to work open, which means they teach you how to build, tools for collaborating with people, for, offering value on the internet, something of value. So they have a lot of people working on different social problems. They have people working on education, open science, things like that. And it's a really interesting set of things that you learn in that group. It's definitely something you can't learn anywhere else. The GitHub repository is here. So it's OWDW Education. If you go to the DevoWorm GitHub, that's what it's called. And it's just a bunch of general education resources. Just different, takes on, like we have a developmental data science resource, which is laying out developmental biology as like sort of a data science endeavor. There's something in pattern formation, something on dynamical systems and development. I could see there being like future resources on machine learning, although that would take a little bit of doing. We talked about this in our group as well, but this is open for, if people want to expand this curriculum, that would be great. We could work on that. Then in the summer, we had Google Summer of Code. And this is, of course, something that's annual. And we've been running this theme the last couple years on machine learning and data science and developmental biology. So this year we have 2 projects in machine learning and data science. We'll have this, what I call a robust application period, which is going to go from February to April, where people are going to try to write up proposals that, address the issues that, so, we were talking in the meeting before the end of the year on proposals that we pose to the general public. And it's, a page long, a couple paragraphs describes the project. Now, what the students are going to do is they're going to turn around and write longer proposals and how they're going to solve that problem. And then we select students from those proposals. But the application period is important in and of itself because it's, it gives people a chance to develop their skills in writing a proposal, but also sort of firming up how they're going to approach the project technically. So this last year we had 2 groups. Vinay, who is actually funded by Google Summer of Code, and he did the semantic segmentation of the embryogenetic process. That was the name of the proposal that we posed to Google. And then the students, including Vinay and including Ujjwal and Asmit, wrote proposals that attempted to solve that. In Vinay's case, he solved it. He used some machine learning and deep learning techniques, and he created a web interface for analysis of data and also a standalone program for it. So, he was able to get a lot of what he wanted to accomplish done out of that proposal. Ujjwal and Asmit wrote good proposals, but they didn't quite make the cut. So I invited them to come into the group anyways and work on a different project. And this is something that Richard had an interest in, which is digital basilaria. So he said, well, why don't we do this digital basilaria project? And I said, okay, I'll see if Ujjwal and Asmit want to work on it. And so they ended up working on it. And they ended up doing a similar thing to what they proposed to do with C. elegans data on the basilaria data. And that's where that paper that I flagged in the timeline comes from. And we also worked with Thomas Harbeck, who's from Germany. And amongst all of us, we ended up, and I think Vinay was involved as well, we ended up putting out a paper on the topic. So a lot of things come out of Summer of Code that aren't just related to Summer of Code. Finally, of course, DevoWorm ML, and I think all of you were here for that, so I'm not going to go through that too much. But just to say that I think it was pretty successful, and this is actually the wrong repository for this, but I can fix that in the slides. It's actually DWML or something like that. But yeah, this happened at the end of the year, and I'd really like to revisit some of the stuff that we presented in that session, or that set of sessions. So in terms of papers, we have 3 things that are kind of going. Raising the connectome, which is something that I've largely been involved with for a special issue of Frontiers in Cellular Neuroscience. I don't know, I just submitted it in the last week, so we'll see where that goes. Towards a digital diatom, that seems to be at a point where we can get some feedback, and that's going to be in a book called Diatom Gliding Motility. So that's similarly in the works, but also in preprint format. And then data theoretical synthesis of the early developmental process. And that was submitted to GigaScience last year. And I got an email from the editor, and they said that they're still working on the reviews. So we'll see, maybe in this coming month or so, we'll hear back about that. So resolutions to do, and this just isn't really resolution so much as what's to do maybe in the next year. I wanted to highlight some things for people to think about. The first is this OpenWorm Docker container. So I don't think anyone here is actually knows what this refers to. Maybe Ujjwal, but so OpenWorm, we had the open house in 2016, and now we're trying to put together a Docker container which contains, like, it would be kind of like the open house where you'd have a bunch of the different simulations that are involved in OpenWorm, all the projects, but in a Docker container that someone can download onto their laptop and open up and run. So Docker is a platform that installs a virtual environment on your computer. So it's like a virtual machine, but it actually is a little bit more user-friendly in that I give you a Docker container, and if you have Docker on your machine, you can open this thing up and it'll execute a bunch of code, and it'll basically reproduce what I want you to see on your computer. So it'll run all of your programs that are in the container. So the cybernetic simulation, the Geppetto simulation, etc. And they'll all run in a way that's reproducible. So if you have a different machine than someone else, you'll see the same content. It'll work in the same way. So this just produces animations for these multiple sub-projects. Right now it's available on Linux and Mac, and they're working on a Windows version, but the idea is to have it freely available for, if you're interested in OpenWorm, you can see all this stuff pop up very quickly. And I think it's a very good way to introduce people to OpenWorm. But the problem is that DevoWorm isn't really represented in this. So that's where this resolution comes in, is that we're trying to figure out how to make a simulation for DevoWorm that's highly visual, that's sort of a simulation that people can run in this Docker container that's representative of, I guess, worm development. And so I don't know what the best way to do that would be. We don't have any native simulation package that would be suitable for this. I mean, like Cybernetic is basically, that whole project is just a software platform. So it's a very easy thing to put into this Docker container. But for DevoWorm, we don't really have anything that we can put in that's just kind of ready to go. So it would require like some coding and some planning. Well, maybe we can make a contribution to future versions of this. So that's something that I've been thinking about maybe for the next year. Another thing, of course, is the Summer of Code, which is happening this coming year. It runs from April to August, but of course the application period starts in February. And this year we have 2 projects that are submitted to INCF, which is our managing organization. One of those is on pre-trained models for developmental neuroscience. So we talked about the pre-trained models in DevoWorm ML last year. The developmental neuroscience part is because we have to make it neuroscience-y for INCF to sponsor it. But otherwise, it's really just about development. And there's a deep learning infrastructure, and the idea is to create an open model for data analysis. And then, of course, we also have the Open Devo Cell Integration Project. And so this is, again, something that follows up on what Vinay was doing. And Ujjwal is on the case. He's working on, a version of this. So, we'll see. I can't promise Ujjwal that he'll get the slot, but I can't do that because of the program. They don't let you do that. But I'd like to see applicants work out the problem, and then we select usually the best person for that. There's a group of us in OpenWorm who evaluate the proposals. So good luck, Ujjwal, on that. Not supposed to put my thumb on the scale, but then of course we have additional data science infrastructure that we can follow up on. We have something called the DevoZoo. I think I've mentioned this before. There's a link from the website, and it's image data and models for embryogenesis and neural development. And that's something maybe we can develop more this year with more forms of data. Um, and, maybe add features like, tutorials. I've talked a little bit to Jesse about, education and how do you like learn these types of skills that you might need for doing like, data science on biological images. So we might have a conversation or two about that on how to implement those things. Um, Comparative development. So we have a lot of, in the DevoZoo, we have a lot of different types of data from not only C. elegans, but C squirts, which you see here in this ball of cells. This is a very early embryo for a C squirt, and other embryos and other developmental processes as well. And so there are a lot of opportunities for comparative development. And I talked to Richard and I talked to Susan about getting axolotl data. So axolotl is an amphibian that we can that has a very easy to view developmental process, and so that's something else we might consider. Highlighting model systems or looking across different systems. Um, and then, of course, this ties to the larger open-worm data science initiative. So for those of you maybe who are interested in things like movement in C. elegans or other things, we have other initiatives that focus on data analysis and data science. So movement database is one thing that OpenWorm has as an infrastructural tool. We have a lot of videos in that database on movement of C. elegans. So they have different mutant phenotypes or, different modes of movement for, um, just across all different types of conditions for C. elegans, experimental conditions. So those things can be mined for, data analysis, for analyzing the movement, things like that. So there's a lot of data there. We also have a lot of interest in building an data science infrastructure where we have like standardized file types and things like that. There's a lot of in the weeds data science type stuff that people are interested in as well. So that's something we can tie into. Also, merging data science or data from biological systems and artificial life. So creating generative models and merging that with data sets. So I told you about Morphozoic, which was the platform that Tom Porteges developed. But, that's like one instance of like a sort of a generative model that we could use to represent development. And maybe even, Morphozoic as being like something that you can put data into more effectively to get out simulations of these type of systems. And so that's another angle that we've not really followed up on in the past. And then I think this is the last slide. We talked about virtual reality very briefly last year, and like I said, I have an interest in virtual reality, and this has a potential tie into the Docker visualization. So this is something called the Worm Viewer, and this is something that OpenWorm has for the adult. And so if you go to the OpenWorm website, there's a little worm at the upper right-hand corner of the website. And if you click on that little worm, it looks like this. You go into this thing called the Worm Viewer, and it's a 3D model of the worm that you can rotate, and you can drop all the cells and the cuticle out of the worm and just look at different cells in their location in the body of the worm. So, you can do experiments where you just look at some of the neurons. You can look at take the cuticle, this skin off of the worm, and look at what's going on inside the worm, different muscles, groups, things like that. And so it's a very nice model of the worm that you can that's layered, where you can take things out, put things back in, by clicking buttons. You can rotate it, you can zoom in and zoom out. That's a nice model of the adult, but we don't have anything like that for the embryo or for developmental worm. And so this is of course a three-dimensional model. So this is something that could be exported to a virtual reality environment where you have goggles and you have, this, maybe you can reach out and touch the worm even. That would be a virtual reality implementation of this. So there are a lot of opportunities for making this virtual, as they say. And, there's, of course, there are 3D, 360 movies now where you get one of these cheap set of VR glasses that you can put your phone into. And then you go to one of these movies on YouTube. They're called 360 movies, 360 VR. And they're projected, they're set up in such a way so that when you set your phone into these goggles, you can see, like in stereoscopic vision, this movie. I mean, virtual reality has a lot of tricks like that they play to project things into your field of view. So there are a lot of things that they don't. They seem like they're magical, but they're really not that hard to implement technologically. There is also the creation of 3D objects like this. This was created in Blender, so it's not too hard to create. We can create 3D infographics and data visualizations as well. And then CompuCell 3D models. So when I was at Indiana University, I talked to the people, and we were talking about CompuCell 3D, which is a platform that they have at their institute, and they developed it there. And it's a model for, they call it a cellular POTS model, but it's basically a variant on cellular automata. And you can model cells in a population of cells like this. This is actually an embryo that they're modeling. But you can also go inside the cell and model gene networks, genetic regulatory networks, and physical properties of the cell. So, like forces and things like that. They typically use data from different sources to build these models, but they can be built sort of de novo as well. And so these generate 3D models of the cell or of an embryo or some biological structure like an organ. And so we never really got into developing anything with it. But if people are interested, it's something that can be done. It's something that can be put into this category. And, maybe it'll lead to something interesting. So new questions to explore. So I talked about technology, but there are also research questions, and I won't get into that now, but we can think about what kinds of new questions you might explore just in terms of the development. So if we have access to data, we know how to analyze data and model things. What kinds of questions can we explore? And I'm sure that there's a lot that we haven't even thought about in our group. So with that, I'd like to exit my screen share, first of all, for now. And so does anyone have any questions about anything let's see, we have some things in the chat here. So, link to blog post. I'll send that out in an email after the meeting. So, Ujjwal, can you elaborate about CompuCell 3D? So I guess, so it's a modeling software. It's written in C++, and there's a developer community where people have developed these models. They've developed them for papers, usually for academic research, but I don't know how easy it is to use really once you get into the weeds on it, how it is to build a model. Okay. But it's basically where you can take, they have these building blocks, so they have these cells that you can build. You build them into a structure, and then you specify what's inside the cells. So, the cells can just be nondescript cells, something that doesn't have any properties at all, and you just simulate maybe a tube of cells, maybe it's some sort of vein or artery, or it's like a liver. I think I saw a model of the liver when I visited them. It was, just basically where you have the compartments of the organ and the cells, and they're doing some function, and you can use real data to calibrate everything, or you can generate your own data to say, well, this is how we think it works. So it's very flexible. Now I don't know how easy it is to take one of those models and like, put them on, put them into the Docker container. But that's not really that's not the first priority for that. The first priority would just to be like to create some developmental model, maybe of an embryo where you have cells and we can put in properties of the cells, like the physics of the cells or the genetic regulatory properties of the cells, and we can like work up from there. We have a lot of sources of data that can be incorporated into these models. So that's why I'm excited about it, because I think we're at this point now where we have a lot of data floating around, and it's a matter of like, putting it into something interesting that people will, really be able to interact with. Dick said, I would like to present on the flipping microscope. Yeah, I was going to put that, we're going to get to the schedule in a minute here. We'll put that on there. I would like to see that as well. I think that would be interesting for the group. Jesse said, I'm interested in the Docker project, education, outreach, of course, and also some things related to multi-scale generative model, data science, artificial life item. I'll have to check out the cybernetics platform as well. Well, it's cybernetics, so it's, okay. We'll go to so it's spelled like that, and I'll send out that link. So let's move on to the agenda, or just to the scheduling of the agenda. So getting up towards the top of the hour, so I don't want to overboard here. But yeah, it's just stuff to think about. Make the slides available after the meeting. And of course, people probably have things to add. Can people see my screen? Okay, so this is our weekly agenda. I just put it up by week going up to the end of May. I just wanted to lay it out. We may or may not meet every week depending on schedules. For January 27th, I put in Devansh and Yash. They're going to do a presentation on deep learning architectures. So we already scheduled that from, I think Devansh set the time. So Dick wants to do something on the flipping microscope. So what would be a good week for you to go?
Oh, anytime.
Okay. Are you like, is it something you can just present like?
Well, I just want to show the paper. It'll take 1 minute.
All right, let me stop sharing here.
Okay. Select. There. Too many screens to choose from here. Window, dictionary. Okay. Now, I know what you can see. Can you see my screen?
Yeah.
Okay, can you see this paper?
I can see your browser.
See my browser.
The browser window. So it's the Meet browser window.
Oh.
Why it's coming up like that, but
Okay.
it might be if you share your entire screen instead of the window.
Okay. How do I do that? Options. Full screen.
Okay.
That work? No, that doesn't work. Oh, that's full screen for me. Exit full screen. Many choices here. Setting before. No, that's not working. Okay, let me stop and start over here.
All right.
Okay, present now. I have two options, your entire screen and window. So I'll try the entire screen.
Yeah.
Okay. Now, I get another window. It says, will you allow me, Google, to see your screen?
Yes.
It says, window or screen to share. And I have about 30 options.
Oh.
I don't know which one to choose. That's the problem. Oh, screen 1, screen 2, screen 3. Not that I'll try screen 1 and see what happens. Ah, maybe we got it.
Yeah.
Wow. That was down at the bottom.
Okay. Yeah.
Now, get the paper. And here
There
it
we go.
is. There, this one.
Yeah.
Okay. So, the idea here is that we're dealing with axolotl embryos, which are roughly spherical and they're bottom heavy. So what you do is you turn them upside down with this flipping stage, and then they right themselves. They turn themselves right side up. Okay, and this is an example. Let me enlarge this. Okay, this is an example. The embryo is upside down, and you can see it's rotating, and it takes a few seconds to rotate. And we take pictures as it rotates. Okay, so the problem is you can produce lots of data like this. The problem is to create a three-dimensional surface view of the whole embryo from these flipping sequences. Okay, and we can flip it in different directions so it writes itself in different directions. So basically it's a montaging problem to produce a map of the whole surface of the embryo from images taken from these different views, which are generated by turning the embryo upside down and letting it turn itself right side up. Okay?
Yeah.
And it's a very explicit problem. I'm not sure what the best approach is. Maybe machine learning algorithm or something. Well, we need something that can match the images and then produce this montage of the whole surface. And it would be a very useful program to have. Okay?
Yeah.
This paper outlines the need for it. You ever have this paper? It's from that Biosystems.
Yeah, I can provide a link to the.
Yeah, I think you've got a copy of it. So
Yeah.
Link to it. So if somebody's interested in working on that, it'd be great to do. Susan Crawford Young is producing the data. She's sending a disk full of data to Bradley right now.
Yeah.
It would open up you see, it's actually four-dimensional data because this is developing over time. We would then be able to track all of the events on the surfaces of this embryo versus time at the cellular level, if we could do this. And there's all kinds of analyses that would follow once we have the ability to produce these montages. Okay, so that's what I want to get said.
Okay.
Okay.
All right.
Yeah. Want to jump into that one? It'd be wonderful to have you.
Yeah. Yeah, I think, we might present it, like, once I get the data, maybe from Susan, we can present something a little bit longer, maybe discussing, like, what.
I think it was drag Susan in.
Yeah.
Okay.
All right. So, I'll put you on the schedule at some point. We'll get it on the schedule at some point. We'll
Okay.
let me get the data first.
Brad, they have another meeting on the hour, so I'm going to have to leave in a minute.
All right, talk to you later.
Okay.
All right, bye.
Okay, thanks.
All right, thanks.
So, start presenting.
So I guess I'd ask, does anyone else have any questions, or is anyone else interested in presenting for a certain week? We don't have to make a decision now on the presentation times, but if you just email me, we can put it into the schedule. So as you saw, we have this empty schedule. So we have a lot of open dates. So if you're interested in presenting on something, please let me know. there's going to be a lot of stuff with maybe summer of code or whatever. Yes, Ujjwal wants to present on DNA fingerprinting. Ujjwal, do you have a date in mind for that, or is that something you might want to do in the next couple weeks? I can put you down for the 3rd of February if that's okay. I mean, you're not like, I don't hold you to that time, but it gives you a couple weeks to put in like some, get the presentation together. So, and then Jesse, I think we talked about doing some, some presentations of papers, some shorter presentations. So we had a couple of papers that we talked about. And we would probably just do that in session here, just to save time. I don't know when would be ready to go on that. Maybe in a couple weeks. Do you have any thoughts about maybe in February sometime? I mean, we don't have to make a decision about that now, but so anyways, I guess we're at the top of the hour. Thank you for attending. If you have any other, I don't know if anyone has any other questions that they want to bring up, or we can wait till next week. I'll send out an email going, with some of the links from today's materials. So, and then next week we'll be meeting again. I don't really know what the topic is yet, but we'll come up with a topic. And I'll send out an email on that as well. Okay. All right. So if there are no more questions, thanks for attending, and talk to you guys later. Have a good week.